Hi, everyone. Thanks so much for joining our talk today. The talk is titled Enter the Nicosystem, affecting the digital transformation of biospecific interactions through DMF, web tech, and a bucket of algorithms. My name's Soleil, and Adam will be joining us for the second half of the presentation. We're both product managers at Nicoya Life Sciences, which is a biotechnology company located in Kitchener, Ontario, Canada, that's focused on building solutions for users who want to measure biomolecular interactions. So the agenda and breakdown of the day. So obviously we're going to focus on all of the ways that we've infused automation into the measurement of biospecific interactions with our platforms. So the first thing we're going to do is just talk about the researcher journey with the data type. And I'm going to sort of orient everyone on what it is that we actually build so that by the time Adam starts talking about how we automate that, we're all on the same page. So the 2nd through 6th points are really going to look at the steps of the user journey. So designing your assay, actually executing the experiment, looking at the analysis, and then really zeroing in on what those results mean. And finally, we're going to have a little bit more forward-looking fun where we talk about some solutions that are coming to you soon. And so as we enter the Nicosystem, which is really just a term that we use for the solutions that we provide our users and the ecosystem in which our users use those solutions, what they're really looking at is the biospecific interactions. So as you can see below, biomolecules are typically reacted in pairs in some condition. And our users want to answer questions about the strength, specificity, and speed of interactions. And why might they want to do that? So all of human physiology is basically one big binding party. Everything that happens, whether it's a disease mechanism of action, just a typical run-of-the-mill reaction in your body, or the way that we understand how a drug interacts with a potential target, these are all predicated on binding events. And so at Nicoya, as I mentioned, we build platforms, which basically just means an instrument, great software, and consumables to enable our users to measure these biospecific interactions. And so everything that we're trying to do is really trying to mimic in some kind of way a physiological environment. And one of the things that you need for that is some kind of microfluidic system that delivers samples to a sensor. And so one of the things that's really revolutionary about what we're doing at Nicoya is that we use a liquid handling technology called digital microfluidics. And this sounds a little scary, but it's really not. It's just a way of manipulating nanoliter sized droplets using an electrode. And so basically what you do is you have an electrode array underneath the droplets, which are suspended in a cartridge fluid or a medium. And these droplets are attracted to when voltage is applied to an electrode. So you can see that's highlighted on the left. When things are red, so when those individual electrodes are red, we're representing that they are engaged, and that brings the droplet somewhere. Now we can also split, mix, merge droplets, but the important thing for our users is that we can bring the samples to the sensor in a way which gets them great data, which is shown on the right-hand side. So there's some fancy algorithms that are applied, but really what we're looking at is a time versus response. And that's what DMF enables us to see, is what is happening at the sensor surface with a myriad of different types of reagents and sample droplets that are brought to the sensor tip. And so how does this actually look on the cartridge? So this is our liquid handling technology. Those are the dispenses. So after something is dispensed from a well, which you pipette it into, they're brought into the mixing zone, and we can perform automated serial dilutions on the cartridge itself, after which the samples are brought up to the sensor, and we expect to acquire our data if there's a binding event happening at the sensor. And how do we know if there's a binding event actually happening at the sensor or not? Well, this is where we get our signal. So in our take on the technology, we use digital microfluidics to move the reactants and the reagents necessary to get good data to and from a fiber optic based sensor. And these sensors use a phenomenon called LSPR, or localized surface plasmon resonance, which utilizes nanoparticles as a substrate on a fiber optic tip. And what this does is essentially enable us to measure changes in mass at the surface in real time. And so what you're seeing is a shift in absorbance. And what our users see, which is important to them, is a magnitude shift in the response at the sensor, which would indicate that binding event actually occurring in real time without the use of labels. So what do our users want to know about this data type? So they understand that there's a response happening, which is representative of a change of mass. And then they understand that there is some time progression that occurs throughout the experiment. But the first and most important thing is specificity. Is there actually an interaction between these two biomolecules? And if so, does it depend on the experimental conditions? The second thing is the strength. So how strong is this interaction? And typically this is reported as KD or affinity. But for us, it's really the ratio of the association, so the forward rate constant and the reverse rate constant. And so that's how we acquire our KD. The great thing about SPR versus typical endpoint assays is that you actually understand how quickly that interaction happens. So both on the association and dissociation side, because we are seeing these events in real time, we're able to understand how quickly they come together and how long it takes for them to dissociate or come apart. And so how do our researchers reason about their work with our platform? Basically, they start with some biological question. Does A bind to B? And if so, how strongly? And if so, how quickly do they come together? And how long does it take for them to dissociate? So the second thing they would do is design their assay. So you'll have one binding partner on that sensor, you'll have another in solution. And Adam's going to talk about this in a little bit more detail, but I'll leave it at that high level for now. And then this assay design that they build in our software, it gets mapped to the biosensor. And the biosensor really is meant to do the work for you. And finally, we get our results. That's where things are analyzed. We have some really cool ways of automating this analysis process step, which can take a little bit of time as you're running through a lot of different experiments. And so just zeroing in on that biosensor step, I thought it might be helpful to look at how this works. So they design their assay on the software itself. That is then shared through the cloud to the instrument, and the instrument is then outputting the data back to the cloud into the analysis software. So the Nicosystem is our user portal where both the design and the results live, and users can easily interact and exchange between the two. So the device operation itself, we really try to automate as many of the steps as possible, which means reducing the hands-on time as much as possible. So let's just take a look at what happens here. So like I said, we produce a platform, which is an instrument, a software, and a consumable, which you see being opened on the screen. And this consumable is what we call a cartridge. And a cartridge is basically just a container for sensors and your different reagents. And that's where you can see the little electrodes that actually are able to move the reagents that you pipette into. And now we're opening the cartridge fluid, which is the medium in which the samples are suspended, as well as your reagents. And so you simply open up this tube and you place it into the cartridge, and it automatically dispenses evenly throughout the entire cartridge. And again, this is building the medium in which you will pipette your samples into. And that makes it so DMF is able to apply the voltage to bring your samples from where you pipetted them up to the sensors that are closer to the top of the cartridge. And now we're looking at our sample map. So after we designed our experiment, Alto tells you exactly where to put what sample. And this will depend on when you want that sample to go to the sensor and how you want it to be treated as it travels to the sensor. Do you want to perform automated serial dilutions on it, or do you just want it to go straight to the sensor? Is it a buffer? Is it a regen solution? All of these things are programmed into the experiment itself. So by the time you get here, you're just following a sample map and inputting the different reagents. And then you start. And Alto closes its door, and your experiment is executed from there. So we truly tried to build a walk-up instrument that was extremely user-friendly. And now I'm going to pass it over to Adam to sort of unpack the user journey for us and look at each of the different steps and how we thought about automating each of those steps. So thanks, and I will hand it over to you, Adam.
Awesome. Thanks, Soleil. So this next section, we're going to talk about how we are automating the first part of the user journey, which is when we are designing assays and mapping them to all the different bits and pieces that need to run behind the scenes to kind of organize information that we get from the user and group things into pieces of repeat work that the device needs to perform. So when we're speaking about everything that happens on the device, we've designed around the particular problems that our users face in performing biospecific interactions. So just a couple helpful terms that we can review today that we're going to generalize for this discussion when possible. So biospecific interactions are comprised of two or more reactants, one introduced from the surface and one from solution. There's usually a condition involved. This is the environment in which the interaction takes place, right? This is a biophysical characterization technique, so temperature matters. Anything else involved in the interaction matters, like the pH, the ionic strength of the reaction's environment, and any kind of cofactors. There's some jargon that sneaks in here in this particular industry. There's the ligand. That's basically what we call the molecule that's on the surface of the sensor that we need to kind of stick around while we bring in another molecule to react with it. Then there's the analyte. This is the solution reactant. This is what comes in the drop that we want to react with the molecule that we've captured onto the surface of our sensor. So we've looked at our main interaction components, putting it all together here. You've got the solution sample, the surface sample, and then the condition. This is the environment in which the reaction is performed. So any reaction that we perform on a device like this or any other can be described as a matrix in n dimensions. So a single interaction requires us to deliver a specific sequence of drops to a sensor, which includes one of our reactants. There's a lot of different things that we're trying to do on this device. Sometimes we're trying to put something onto the surface of a sensor. Sometimes we're trying to react something with it. And other times we're just trying to modify the environment in which the reaction occurs. We are conditioning the molecules on the surface or performing some other action like that, or allowing them to stabilize over time. Pretty much all of these behaviors can be described as sequences of drops, basically the steps that we need to perform for any type of activity that we perform on the device. So a couple examples here. What we do is we allow our user to describe their experiment as simply as possible. So for example, I have M surface reactants and N solution reactants. In this figure here, you can see that we've got our M and N. Those are the numbers of things that we're trying to react, which define this matrix, which we then scale oftentimes by concentration. So that's already 3 dimensions that we need to accommodate on this particular device, in addition to all the other ones that we can scale from here. For example, like we mentioned, pH, ionic strength, and so on. And then you can see kind of our first example of the output data type, which is the sensogram, and a helpful visualization across the top of the sensogram, which is the drop timeline. So you can see the drops arriving on a particular sensor, and then the output signal from that sensor. Some pretty common topologies that we encounter in these matrices can sometimes be described as one on many, many on one, but very commonly many on many. And these are just a couple examples of what our researchers deal with. So in the one on the left here, we have a lot of different reactants coming in from solution. And then we have one, for example, particular protein that we want to react with that we're going to place on the sensor surface. Then in the next example here in the middle, you can see that we have just a few reactants from the solution phase, but we have many that we want to place on the surface of the sensors. The reasons for doing this we won't get into in this particular conversation, but they often have to do with valency and the stability of the molecules that we're working with. And finally, on the right-hand side, you can see there are very many, in this case, compounds that we're introducing to a few proteins that we've put on sensor surfaces. So we can basically extend this framework to accommodate any interaction system a user is interested in with just a few inputs, right? We've described how many things we want to react from the solution phase, how many things we need to put on a surface, and then any other conditions that might be involved. So what this does is it tells us how many unique sensor addresses we need. It tells us how many unique wells or physical addresses we need to put unique samples into a cartridge or across many cartridges in an experiment. And it also tells us how many conditions we need to add into the cartridge. So, and total volumes that we need to accommodate overall. So this is the basic framework that allows us to kind of scale any experiment that the user could possibly design around this data type. And we've built tools that allow users to expressively describe what they want to do on our devices. You can see here on the left-hand side some assay templates and things like that for selection. And on the right-hand side, the basic inputs and forms that a user has to help define what they're trying to do experimentally. This is a figure I particularly like. So the left-hand side is the tools that our users have to work with in software. But on the right-hand side, we're visualizing how the data structure scales behind the scenes based on what the user is trying to do. So our software takes all of this information, and we've designed algorithms that help the user get the most out of the hardware that they're working with, right? A lot of times things can be done in parallel. Other times it's better to do things serially. It just depends what the user is trying to do, and we built algorithms to accommodate all of that. So as we map the data to the hardware, this is the final and really important step of this stage in assay design, where we need to not only set up what happens on a single cartridge, but very often the experiment that a user has in mind might require more than one, for example, input traditionally. But in this case, because our entire biosensor is basically defined by the cartridge, their experiment might need to scale over multiple cartridges, and so our algorithms accommodate that as well. You can see in this example, we're scaling as we tweak the dials here and define our experiment. You can see it scale within one cartridge, depending on what the user's doing, and then over multiple cartridges. To our ecosystem, this is still one experiment, but it has already been broken up into multiple methods that can be run on a single device in series or across multiple devices if you have them in parallel. All of the data will be analyzed in pieces as it is available, but the final report can be generated from the set in aggregate. Okay, so that's how we automate the assay design. Now we're going to talk about how we automate experiment execution. So this is one of the coolest pieces of this technology, and all of the work that we've seen so far in automation, it really is enabled by these techniques that we're going to talk about here. So the first piece is we need to break down our experiments into activities. This is a logical model that we, or a logical grouping that we use to describe work that is performed routinely across many different experiments. So very often we're trying to build a surface. For example, we want to put molecules on the surface of our sensors. And in this case, you can see here, we've just got basically the most simple activity you can possibly think of, where we've got one molecule on the surface reacting with one in solution. And we've got what we would call our interaction object and associated results here on the right. So that's high level what the user wants to know, right? They want kinetics, right? And that's an activity designed to get them kinetics. Within an activity, we need to perform a number of drop sequences. So this is an example of what a full experiment would look like, where we've got the activities across the top, and you can kind of see the hierarchy of our data structure, the cycles that we group them into, the sequences, and then the sensors that those sequences are assigned to, right? And so if you kind of look at the insets here, you can see here's still our simple one-to-one or one reactant in solution, one reactant on the surface model, but you can see all of the steps that are required to perform this basically, right? You can see all of the 5 sequences grouped into the 5 cycle, sorry, the 2 sequences per cycle required to generate the data and have a reference channel basically. And then you can also see the surface building steps and a couple other support activities like normalizing the cartridge and cleaning the sensors. All right, this is the coolest part, I think, of the technology. Everything that we're doing is basically we've quantized this entire data type, right? So there is a discrete unit involved for every different step that we need the device to perform. So everything basically breaks down into drops. So here we have an example of the 5 different drop types, and I don't think you can see them. Let me see if I can move this screen. There you go. So You can see here we've identified 5 major drop roles. So anything that the user puts in the cartridge probably falls into one of these 5 categories in terms of the experiments and what that solution is going to be used for. So I'm just going to move this back right here. There we go. Okay. And you can see this example here where, like we saw earlier, we're moving drops towards a sensor in order to execute that drop sequence. So you can see the drop sequence executing here, and you can see the data being made available in real time as the drops are addressing the sensor. Unfortunately, I did not have time to synchronize this figure, so they're a little out of sync, but you get the idea if you let this cruise for a little while. All right, so let's keep on rolling here. All right, so all of these core behaviors are enabled by DMF. So what can DMF do for us? The first thing that we can do on our devices is dispense. That's where we take from a common reservoir some number of drop units and dispense them onto the transport layer. Now, drop unit is around 300 nanoliters, and so we can distribute that volume or larger trains of drops of many drop units around the cartridge. The next step is moving them. Once we've dispensed into the transport layer, the main operation on the cartridge is really just moving drops around the electrode map. And finally, we can split drops. You can see in this figure, we've got a 2-drop unit that we're splitting into 2 drops, 1 drop unit each, basically. All right, so some advanced operations. We've got shake. So sometimes we need to mix a drop, or to introduce good transport conditions, we want molecules to get to the sensor surface so that they can react. So a lot of times we try to induce good transport conditions to minimize the height of the boundary layer by oscillating them on the surface of our sensors. Then sometimes we also need to mix things together. This is super helpful on device for mixing activating reagents that have a short half-life once mixed, or a short working time. We need to use them as quickly as possible, so that's another useful operation that we have on device. And then super advanced operations are these choreographed subroutines, right? So let's say we need to perform an automated elution where the user actually needs to introduce a train of drops to the sensor surface, and in their drop sequence, they need a serial or geometric dilution series, right? And in this case, we can actually, you can see we're introducing a stock concentration. We've arranged the stock concentration and then a set of 5 buffer drops, so no concentration equals 0, and we're just kind of passing along a 1-drop unit to all these 2-drop unit drops, which performs a 3-fold elution series, which is perfect for performing kinetics, and that's exactly the type of thing that certainly from a personal experience, try not to think about how much time I've spent making dilution series. So this is something really cool that the device can do for users ad hoc. All right. One more thing to talk about here is that all of this that we've just looked at, all of these cool DMF subroutines and the framework for how we talk about the hardware as sets of or maps of sensors and a transport layer and a sample layer with a bunch of unique addresses to put unique reagents in, this experiment description as a set of drop sequences that are arranged in cycles and activities and so on. This is a platform that we can build on. So we have a ton of freedom on this device to continue to make new cartridges and fundamentally adapt to specific applications. But nothing really changes around these core, the core engineering of the device. So we still have an instruction set, we still have a cartridge map, and we still have an experiment. So what we can do is with this information, we can calculate all of the electrowetting commands that we need to perform the experiment, which is very cool, and which you might consider an ahead of time or AOT compilation technique when it comes to the electrowetting. But a big question that some of you are already asking, I'm sure, is can this device be programmed to make decisions in real time? There are lots of scenarios where, let's say you have a concentration, but you have an interaction you need to perform, but you're not sure what the KD is. what concentration do you use? Typically, you would optimize and you'd try it first, and then you would kind of figure it out, and then you try to bracket the KD, or you just run everything with like a at least a high concentration, just kind of see what you get, and then maybe tweak it later and rerun things. Or let's say you run something and you didn't quite optimize the regeneration condition, and so you've got incomplete regeneration. What do you do, right? Can the device help you out in these situations? Of course, the answer is absolutely yes, as you might have guessed. This is very much the promise of digital microfluidics combined with optical biosensing. So we call this, there's a lot of tech terms here, but we'll just call it a kind of advanced scheduling or dynamic scheduling. What that means is we're able to use this kind of dynamic discrete event simulation routine to take what needs to happen in context with what has happened and figure out what needs to happen next, right? So a lot of the problems that this helps us solve specific to our current users in the optical biosensor space, precision target signals, right? A target is reached, do something. Target is not achieved, do something else, for example, like time out or give up. Affinity estimations, let's say we're able to make one really quickly with just like a quick drop addressed to the sensor surface. We can very easily from there estimate a KD, right? And then perfectly choose the concentrations needed to bracket and basically generate perfect sensograms. Smart off-rate detection, right? If we're really looking to guarantee 5% decay in our off-rate, or if we don't want to waste a bunch of time on a particular interaction, dissociating when we don't really need to, and we've got plenty of decay in order to fit an accurate off-rate. That's another situation where this dynamic scheduling is super helpful. And finally, problem sensor detection. Let's say we have a sensor that gets fouled, happens all the time when you're working with novel compounds. That sensor, typically you would just let the experiment continue. After the fact, identify problem sensors or problem spots within a run, and then rerun those compounds. Let's say you could identify it in real time and requeue those samples to a good sensor, so that you don't have to stop the experiment, and by the time the experiment finishes, you are guaranteed to have good data. So these are situations in which the dynamic scheduling that is enabled by the digital microfluidics really helps our users out in automating a lot of processes. Finally, there's the operation of the device itself. We've talked a lot about what actually happens inside the machine within the context of a single experiment. But let's talk a little bit more about a solution that we've designed for scaling experiments outside of what we can do inside of a single cartridge. We've got a collection or a cluster of 3 robots here. We've got the Alto, we've got a standard PF400, and we've got an OpenTrons, a liquid handler. And so what this setup allows us to do is request cartridges, right? Alto can say, hey, OpenTrons, make this cartridge for me. And it knows what's in the OpenTrons, and also it can control the environment of the samples, not only in the Alto device itself, but also in the OpenTrons, because they have a lot of nice modules for example, sample chillers and things like that, that can help us maintain the environment of samples outside of the Alto itself. Loading and removing cartridges, storing multiple cartridges that might be needed across a large run, pipetting samples like we just talked about into the cartridges, and basically operating itself. So in this way, Alto can talk to these other robots in this cluster and get exactly what it needs to work through a large experiment or multiple experiments in a queue. And then finally, final point in this section is that we absolutely recognize that machines are users too. A lot of the most exciting work, I think, in drug discovery is coming from these automated workflows and platforms in biophysical characterization. So we absolutely have planned support for this fully automated environment, both from experiment creation to device operation, like we just showed you, all the way through analysis with our Nicoya APIs. All right, so now the really fun part. This to a lot of users is the product. Certainly if you're not in the lab operating the devices, this is what you'll see out of this platform, is you'll see data. And so we've engineered some fully automated best effort analysis techniques for across our application landscape. And this is all supported by our cloud architecture. So if you look at the figure on the right here, ignore what's under there. It's basically just our devices in a little row. All this is the takeaway here is that there are, a main web app that we use to host our analysis tools, our design tools, our some dashboards to help users, see what's going on all of their devices, and support tools. And this is where we host a lot of our analytical techniques, but we have also, I'll talk about that in a moment. Let's first talk about the data structure. So core to our data structure are these data drops and interaction objects. This helps us get data where it needs to go and is ultimately highly composable. You can see that we collect data at the drop level. So every time a drop is in contact with a sensor, we are collecting data on that interaction. So at the end of an experiment, we have a bucket of drops. We can arrange those drops based on what the experiment did into these interactions, which are usually experiment specific. Sometimes if we're looking at QC, we'll want to be looking at a particular drop in every sequence. Let's say we really want to look at baseline over the course of an experiment. We can look at all the buffer drops. Every time buffer was in contact historically over an experiment with a sensor, we can group all of those together and very easily look at those without having to look at other things that may not be related to it. But if we're looking at interactions, for sure, we're looking at these kind of like baseline drops, as you can see on the right, the solution sample, and then usually that dissociation phase where we're looking at the decay and the interaction. We can group everything together based exactly on what we need and then perform further analyses from there. Everything that we do in the analysis tools is kind of best effort at first approach. So what this means is, as a user, when you log into the portal, if an experiment is complete, your results are immediately available. We, of course, provide all of these different views that you see here to allow you to go in and tweak data and continue to explore it and reanalyze it as necessary. But we also do the best we can to design the software to direct you to anything that needs your attention, right? So you can filter and group by lots of different tags. The good data, obviously, you probably don't need to interact with, but certainly if there's problematic data that you might want to try to salvage or use in some way that the software is unable to or recommends against, you still have the freedom to do that. All of this is built upon a classification and tagging system. This is super exciting. So if you are not into data science, you probably work with the data science group, I'm sure. And certainly they will have a lot of things to say about this, but we basically have multiple tags available for every kind of experimental feature. Those are the interactions themselves, the samples, things that you care about from the experiment perspective, as well as system features. So sensors, again, let's say a sensor gets fouled. That's something that you might want to know, but is based on the sensor itself then, right? And not the actual interactions. And then wells, drop paths, things like that. And this is kind of an example. You can see on the left here, a couple simple tags. This is a regeneration QC, it looks like. And then over here, we've got some kinetic analyses and a lot of different classifications as well for biphasic interactions, non-binding, anything that we can help you identify, we will do. And you can, of course, overwrite and redefine the classifications as needed in the same set of tools. Really briefly today, just wanted to introduce a couple simple things around some application-specific modules, just to let you know what's coming as we release more and more tools. I think that's one of the coolest things about this web-based analysis package is that it makes it very easy for us to continue to push new features to our users and help them get more out of the machines. So these are a couple examples of some epitope characterization tools that we have coming out. And a lot of, we have purpose-built data visualizations and, again, tools that to try to help you get the most out of your sensograms. And finally, the last point to note on how we're automating analysis is that all of the tools that we make available to support the web app are also available on device. So it's an NPM package that probably doesn't mean anything to most people here, but it can run on the device, it runs in the cloud, it can run anywhere our data goes, basically. So on the device especially, in real time, we can analyze data and help the device make decisions. In this case, the device is telling us that Looks like an affinity is likely single-digit nanomolar. That's great. The off rate looks pretty fast, and we have a 1 micromolar stock in the cartridge. So the device in this case can make a decision to start off at 300 nanomolar and perform a 3x 3 dilution over 5 points, and that should perfectly bracket the KD based on that input information, which is a lot of the information that you might have done in a lengthy kind of optimization process. And then over here on the right, we've got our some notifications saying that analysis is available because an experiment completed on the device. As soon as it's done on the device, you know that in the cloud, and it looks like there are a couple interactions that need review, and it's letting us know that as well. Finally, automating results, getting data where it needs to go. I don't have a lot to share at this stage, but all of this is being built into our Nicoya APIs. We have planned out a REST API and data services that can help our users get data where it needs to go. Let's say you're, you've built out an automated characterization pipeline, and you need, whenever an experiment finishes, you want to set up a trigger to go and fetch that data, or just the interactions, objects, or whatever you need from that experiment to feed your data pipeline. These APIs will help you do that securely and get your data wherever it needs to go, as well as set up experiments and a couple other cool features that we'll talk about in a future presentation. So welcome to the Nicosystem. This is a digitally transformed platform for discovery with optical biosensor data.
That's awesome. Thank you, Adam. And thanks so much, everyone, for joining us today. This concludes our overview of the Nicosystem and the effect that digital microfluidics brings to optical biosensors. For those of you who find yourself studying biomolecular interactions and are looking to transform your journey with label-free biosensors, we invite you to join our growing base of Nicosystem users. For any questions about today's presentation, you can reach us via the contact information that's on this slide, and we really look forward to hearing from you. Thanks so much, everyone, and have a great rest of your day.